Traditional management of panic disorder focuses heavily on the localized synaptic concentrations of serotonin and gamma-aminobutyric acid. While selective serotonin reuptake inhibitors and benzodiazepines target these receptors, they often prove ineffective in stabilizing the autonomic nervous system in treatment-resistant cases. For a specific genetic cohort, the physiological drive behind hyperarousal is a systemic enzymatic dysregulation. This dysregulation exists at the intersection of 1-carbon metabolism and the clearance rates of excitatory catecholamines, specifically dopamine, norepinephrine, and epinephrine. Specifically, variations in the MTHFR and COMT genes physically restrict an individual's biochemical resilience to stress, dictating their baseline autonomic arousal. Mapping this genetic intersection reveals the mechanics of the hyperadrenergic state and the limitations of a purely receptor-centric model. In regions like the striatum, the brain clears dopamine rapidly using dedicated dopamine transporters. The prefrontal cortex, however, is remarkably sparse in these transporters. To regulate dopaminergic tone and clear stress hormones in this region, the brain relies almost entirely on an enzyme called catechol-O-methyltransferase, or COMT. A highly prevalent polymorphism in this gene, known as the Val158 MET mutation, alters the enzyme's thermostability. Individuals homozygous for the MET allele produce a structurally unstable, slow COMT enzyme that operates at a roughly 75% reduction in clearance capacity. This chart visualizes the Yerkes-Dodson curve, mapping cognitive performance against autonomic arousal. Because their enzymes struggle to clear catecholamines, slow COMT individuals naturally maintain elevated baseline dopamine. In low-stress environments, resting neurochemistry sits exactly at the optimal peak of the curve, granting superior executive function. But under acute stress, an adrenal flood of catecholamines completely overwhelms the unstable enzyme, violently pushing their neurochemistry down the slope into hyperarousal. Because the hormones cannot be efficiently degraded, the biological half-life of the fight-or-flight response is drastically prolonged. The lingering epinephrine and norepinephrine sustain an intense sympathetic response, manifesting clinically as severe tachycardia, rumination, and unprovoked panic attacks. The slow COMT phenotype creates an inherent structural vulnerability. Even minor environmental or chemical stressors generate disproportionate and sustained hyperadrenergic states. The COMT enzyme cannot function autonomously. To physically degrade a catecholamine, it must attach a methyl group to the neurotransmitter molecule. The exclusive donor for this methyl group is S-adenylmethionine, or SAMe. This creates a dangerous dependency when an individual also carries a severe mutation in the MTHFR gene, such as the C677T variant. This upstream genetic bottleneck drastically reduces the synthesis of active bioavailable folate. Looking at the folate cycle, we see how an upstream stall in homocysteine remethylation precipitously drops systemic SAMe levels. Without adequate active folate, methionine production halts, starving the COMT node of its required fuel. As the cycle fails, upstream S-adenosyl homocysteine, or SAH, immediately accumulates and directly blocks the remaining functional COMT nodes. Within the cellular environment, SAH acts as a potent, direct competitive inhibitor against almost all cellular methyltransferases. It actively chokes out remaining COMT function. The patient is now subjected to a severe triple deficit. First, a structurally mutated, slower enzyme. Second, starvation of required SAMe fuel. Third, aggressive suppression by SAH inhibitors. In female patients with these genetic variants, autonomic instability follows a highly predictable timeline. Panic disorder and anxiety frequently peak at two distinct points during the menstrual cycle, mid-cycle during ovulation, and the luteal phase prior to menstruation. This 28-day chart tracks estrogen and progesterone against a panic threshold. Mid-cycle, spiking estradiol monopolizes limited COMT enzymes and epigenetically silences new enzyme production, locking out dopamine. Later, luteal progesterone upregulates MAOA, aggressively depleting serotonin and destroying inhibitory control. This intersection of cyclical estrogen surges and progesterone-induced MAOA activity creates a predictable window of enzymatic competition for the slow COMT genotype. Recognizing an MTHFR mutation often leads clinicians to prescribe high-dose methylated vitamins, like methylfolate or methylcobalamin.
For patients who also possess the slow COMT genotype, this approach routinely triggers severe adverse reactions. A sudden, massive influx of methyl donors creates an overmethylation crisis. To maintain homeostasis, the body forcefully upregulates two primary regulatory methyl sinks, the GNNMT and NNMT enzymes. Looking at these pathways, we see that as the methyl sinks activate, they rapidly burn through the available same pool to clear the excess. In doing so, they flood the local cellular environment with massive amounts of inhibitory SAH. This SAH flood paralyzes whatever minimal COMT function remained. The result is a violent, unchecked accumulation of excitatory neurotransmitters, presenting clinically as severe irritability, hostility, and acute panic. A similar chemical gridlock occurs through diet. Many patients unknowingly trigger hyperadrenergic states by consuming high catechol-flavonoids, specifically quercetin and the EGCG found in high concentrations in green tea and matcha. Because these specific dietary compounds possess a catechol ring structure, they must be metabolized directly by the COMT enzyme. Consuming them occupies the severely limited enzymatic bandwidth required to process and clear endogenous stress hormones. Whether through heavy methyl donor supplementation or generalized wellness flavonoids, adding competitive inhibitors to a constrained system actively suppresses clearance capacity, turning a baseline biological vulnerability into an acute crisis. Safely managing the MTHFR and slow COMT phenotype requires a precision approach that utilizes alternative enzymatic pathways without introducing heavy methyl donors. The liver possesses a secondary parallel remethylation route known as the BHMT pathway. This enzyme functions completely independently of MTHFR, folate, and B12. This diagram shows the parallel BHMT pathway. By utilizing dietary choline, oxidized into betaine, the BHMT enzyme strips a methyl group to remethylate homocysteine. This process guarantees a steady synthesis of SAMe, supplying up to 50% of cellular requirements. Despite optimized pathways, extreme stressors can still precipitate overmethylation or active panic attacks. In these acute scenarios, high-dose niacinamide, vitamin B3, serves as one of the most potent targeted rescue protocols available. Biochemically, the metabolism of niacinamide relies entirely on the NNMT pathway. Administering large pharmacological doses artificially forces this pathway to act as an immediate methyl sponge. It rapidly drains excess SAMe out of the systemic pool, dropping SAH production, and halts the catecholamine storm, while exerting anxiolytic effects through indirect GABA modulation. Safely managing this genetic intersection relies on establishing a steady alternative fuel line for methylation, combined with a precise biochemical off-switch to halt panic at its onset. Even with optimized cofactors, a structural limitation remains. The slow COMT enzyme physically lacks the kinetic machinery required to rapidly clear a sudden, massive release of stress hormones. Purely biochemical solutions often lag during an acute crisis. To prevent the initial adrenal release of catecholamines that the enzyme cannot handle, behavioral somatic interventions must be employed as a frontline defense, specifically respiratory vagal nerve stimulation. The vagus nerve physically passes directly through the human diaphragm. Executing slow, deep, controlled belly breathing mechanically stimulates this neural superhighway. Activating this ventral vagal pathway forces a shift in autonomic balance toward parasympathetic dominance. It acts as an immediate biological brake on the sympathetic nervous system, preventing the physiological momentum of a panic attack before catecholamines can accumulate. The successful protocol spans both biochemistry and behavior. It requires removing competitive dietary inhibitors, maintaining the BHMT bypass with choline, utilizing niacinamide as acute chemical rescue, and employing somatic vagal downregulation to control arousal at the source. By abandoning simplistic receptor blockade models and treating the underlying metabolomic constraints, clinicians can stabilize these complex genetic pathways, transforming a highly vulnerable biological phenotype into a state of resilient, high-functioning cognitive flexibility.